So let's dive into the webinar here. So first of all, thank you all for joining this webinar on mastering design protocol figures. As a quick FYI, the entire content of this presentation was actually created in BioRender as well, and we'll be presenting directly in BioRender, so you can get a sense of how that flows. And here's today's agenda. So, after a brief introduction, we'll dive into the most efficient process of building a protocol figure. So, today we're going to be focusing specifically on protocol figures. I'm sure it's something that everyone here is involved with. And then we'll be spending some time outlining really the top 5 tips for ensuring that your protocol figure is as effective as possible. And then we're going to be doing my favorite part of the entire presentation, which is going to be critiquing some figures together. So this is a collective effort. We're all going to participate and critique a couple of figures together. And then after the critique, we'll be sharing a very special announcement at the very end. We think it's going to be a bit of a game changer for a lot of our users, so be sure to stick around for that. Okay, so just briefly, we'll start with a little bit of an introduction. And we'll actually start by introducing ourselves a little bit. So my name is Mina Nashid. I am a scientist. I have my academic background in biology and biochemistry for my bachelor's degree. I did my master's in neuroscience and my PhD in medical science. All my degrees were from McMaster University in Canada. And I am one of the science designers here at BioRender. So that means I am one of the people responsible for creating some of the figures that you see, the figure templates that you see in our library. And I'll toss it over to Sally.
Yeah, hi everyone. My name is Sally Kim. Like Mina, I'm also a science designer here at BioRender. I did my bachelor's and my PhD at University of Toronto here in Canada in physiology. Specialize a little bit on female fertility while doing my PhD studies.
Awesome. Thank you, Sally. So we're just going to be turning off our videos for the rest of the presentation, or at least most of it, just to make sure that we don't run into any technical issues and everything runs smoothly. But we're still going to be back here, so hopefully this just gives everyone a chance to kind of focus on the presentation as well. So before we dive in, we always like to share our mission statement here at BioRender, which is to accelerate the world's ability to learn, discover, and communicate science. So to that end, obviously, we know that scientists often have to communicate about their protocols, and really comprehensively depicting protocols is essential for both effective communication of what you're trying to show in maybe your paper or grant or presentation, as well as for experiment reproducibility. We all know as scientists that this is kind of a big deal in research, is being able to reproduce experiments, and really clearly communicating your protocols is an essential step in that process. So feel free to drop in some answers here in the chat, but we also wanted to open it up to everyone here and ask, what do you typically struggle with the most when creating a protocol figure? So while everyone kind of shares their insights here, I'll share a little bit for me. As you saw, I have a background in academia as well. I was a researcher for over 10 years, and for most of that time, we really didn't have BioRender. BioRender didn't exist. So I know for me, a big pain point was trying to cobble together the images and of equipment from Google. So things like, if specific centrifuges or specific equipment for like animal behavior protocols, tubes, that kind of thing, and really put them all together. In a way that made sense, which was really, really time consuming for me. I also had a really hard time deciding on really how much detail to include and how to present my visual steps accurately. So if I'm being completely honest, a lot of the time I would just, it seemed a little bit daunting for me, and I ended up just relying on the text description without any visuals whatsoever, which made my protocols a lot less effective as a piece of communication. So super glad, I mean, first of all, to be working for a BioRender, but that BioRender exists for all of the scientists all over the world, because hopefully that can solve a big pain point for a lot of us here.
Yeah, just jumping in to say a lot of the comments maybe echo what your struggles are, Mina. So,
Yeah.
some like sometimes like lacking like what to do artistically, like I don't know what to put on, so having trouble with that. Flow of information is another common one I've seen, layout, being concise, choosing what to put on the page too. Yeah, so some similarities amongst the crowd here.
Yeah, I mean it's kind of nice to see that we all share these similar pain points because here at BioRender we're trying to address this for everyone and kind of knowing what everyone struggles with really helps with that. Okay, so if we go to the next slide, you'll see here, let's start with a little bit of an analogy. So let's imagine for a second that we are planning a trip from San Francisco to Los Angeles, and we want to reference some sort of map to make our journey. So looking at these two maps, maybe take a second to ask yourself which one would you choose in order to be able to confidently guide yourself throughout that journey. So which one do you think is more effective in terms of guiding you? And for me, compared to the map on the left, the Google map on the right is better in a few very distinct ways. So first of all, it has a much easier to follow composition. It minimizes the use of color, which is really important here because the use of color is strategic in terms of drawing attention to just the key elements that you need for the trip. So there's, a very clear blue line that takes you through that journey. Everything else in the background is a little bit more muted, so your eye really focuses on that journey that you're making. It also has a very effective starting point and ending point. You can see it's labeled A and B, so you know where you're starting, you know where you're ending and really just highlights the most important information that you need. And similarly, we kind of want to keep this in the back of our mind when we're thinking about building a protocol figure. So when we make a protocol figure, we need to have a logical flow. Everything needs to make sense in order. Um, and we need to have a very clear starting point and ending point with no visual distractions that can create confusion. So just keep in mind that an effective protocol figure is one that really supplements the detailed protocol that you provide in terms of text and provides a clear overview that ensures your audience can really confidently follow along or even be able to reproduce your steps. And I'm glad to see in the chat that everyone chose B as well. Uh,
Nice.
yeah, great. Uh, awesome. And with that, I'm going to toss it over to Sally, who's going to be taking us through how to build a protocol figure efficiently.
Yeah, thanks, Mina, for that great introduction. So yeah, hopefully with what everything Mina has introduced to us, it's clear to everyone why making effective protocol figures are really important. So now we want to move on to the part of the webinar where we give you the tools to help you the next time you make protocol figures, starting with our steps on how to build a protocol figure. So let's get started. So as science designers, Mina and I make a lot of figures, as you can imagine. And over time, we have realized that following these 5 steps really helps to make our figure making as streamlined and easy for us as much as possible. So we recommend that you try out these steps the next time you go about making protocol figures. But of course, let me go into these steps in more detail so you know what each step is about. So the first step, which really starts even before I open BioRender, is to consider the context and the audience that my figure will have. So this is because the amount of information I will have on the figure will depend on what the figure is for and who the figure is for. So I make sure to have thought about both before embarking on the rest of figure making. The next step is to make a reference board. So what this means is collecting all the visual and written material that I want to reference for my figure and add it all to one location. So I like to do this right on an empty BioRender canvas that I will be making my actual protocol file or protocol figure on later. So let me actually do this for you. Let me actually demonstrate this for you. As Mina said, we are working in BioRender right now, so I can do that easily for you. To do that, let me just push this timeline of steps right at the top so you can still follow along. I do want to mention that as I'm demoing, for those of you who joined us today who are not as familiar with the BioRender app, that I will be pointing out the features as I use them. But we also do have a learning hub with a bunch of wonderful educational resources for you. So feel free to access and learn more from those as well. But let me get right back into the reference board. So let's say for the purpose of this demo that I want to create a figure about, let's say, resuspension of a cell pellet after harvesting from cell culture. You can see that I already have quite extensive notes and a written protocol for this within my personal document. Let's say also that this figure will be for my teammates as well for a presentation. So it can be pretty simple, doesn't have to go into much detail. So to build a reference board for this, I will simply start adding all the info that I need onto this empty slide while making my figure, including any text. So to do that, there's a couple ways to do this. I can simply go to, let's say, a Google image search and copy and paste any pictures that I found that I think will be relevant and helpful. So for example, this figure I like, so you can copy, go to my empty canvas and then paste. There you go. With that, I can also resize items as well. Alternatively, what you can do is I can go to the upload section, go to upload and add any other files that I have already saved onto my computer. And once that's been uploaded, I can simply click and then add those to my reference board as well. And again, resize if I need to. And I will say there's a variety of reasons why I'm adding on these particular figures. So for example, this first one that I added here, I like this particular figure because it does depict the actual protocol that I want to illustrate. So it's helpful for me there. These latter two I added, they're not relevant in terms of content, but I like how they look stylistically. So I want to pursue that in my own figure. I can also add in text that I think I will be referencing. So for example, specific protocol steps. I often refer to those when making protocol figures. So I can go to text, create a little text box for myself here, and then simply paste. I can also pull any icons that I think I'll need during my figure drafting. So to do that, I'll go to icons on the left-hand side here, and I can search any particular key terms that I think will be relevant. So for example, I think I'll need some tubes, so I can pull those out and add them to my little reference board. And I can keep doing that until I arrive at stuff until I arrive at a pretty comprehensive place and that I'm happy with. So something that looks like this, like that. So after you build a reference board, the next step essentially is to sketch it out. So this is the step when I consider all the references that I have collected, brainstorm what my figure is going to look like, and then draw it out. So for example, let's say after some consideration of all these reference figures, and text, I think I'm going to base my figure stylistically on this image right here. So I like that it has just a few panels, a few steps, and some arrows in between. So I'm going to also sketch something that looks similar for my figure. So to do that, I'm going to create some empty space for myself on this canvas. To do that, I'm just going to select all the things I've gathered and then push it over to the side. So I still have it close for reference. So if I need to look at anything again, I can just scroll to the side to look at it. But now I do have this empty space that I can, you know, pull things out on. So now I'm going to start building a sketch right here. To do that, what I'm going to do is I'm going to add some shapes. So I'm going to go to shapes, pull out some rectangles, and add these as placeholders for the number of steps or panels that I'll have. I can also start pulling in some placeholder arrows as well, because like I mentioned earlier, I want to have some arrows in this as well. So you can do that, fiddle around with the placement of things, the number of things, until you arrive at something you are comfortable with, something that looks like this. So now with all these placeholders, I can envision more closely what my future figure will look like. And if you're happy with what these placeholders look like, then on top of this, I can start pulling in additional things as well. So for example, I can start pulling in the icons that I pulled from my reference board, start adding it to my sketch to see what that would look like. You can add them some text until your sketch looks something like this. So this way you have a very good idea of what your figure will look like, although it is quite rough, but the major pieces are already blocked in. Okay, so the next step from this point on is to arrange icons and shapes. So this is the point in the figure making process that I will be arranging the icons on here more purposefully than I did in the previous sketching step. So this means, removing any extra items that I may have pulled on during the brainstorming stage, like the placeholder shapes, switching out any icons that I placed during the sketching phase with more appropriate ones. So let's say one that's more indicative of a collective collected pellet, and start changing the sizes of items too, so they fit better within the area I have placed them until my figure is quite close to what I envisioned it, and it illustrates my protocol clearly. So something that looks like this. At this point, all that's left is the last step, of course, which is to fine tune the figure and add any last finishing touches. So this can include things like tweaking the styling of items or the alignment of them. So for example, for this particular tube, I think I want to change the color of this pellet right here. So to do that, I'll select the icon and go to the left panel, open up the panel, and you can see that I have a variety of editing functions available to me. I'm going to choose another color preset like this. That's more indicative of what the tube will look like in real life. And I'll also do a similar thing to this shape right here and choose a more pinkish color. So I'll do that so that everything looks a bit more unified and more closer to real life. I can also make these arrows longer to have them fit better within the space that I've set it for. And yeah, I'll continue tweaking until I'm happy with the results. So something that looks like this. And at this point, I'm done adding all the finishing touches to my protocol figure, and I'm free to do whatever I'd like with it. So one example of something I can do at this point with Premium BioRender, I have access to the share and collaborate feature. So this means I can click share and invite whoever I like to this file by typing in their email. So in this particular case, let me add Mina and then send an invite. So that email has been sent off to Mina. And so then once he gets my email, he can open this file, comment on it, do some live edits as needed.
Yeah, hi everyone. I'm back. Thank you, Sally, for sharing your file with me. I'm still here. Now that I have this file shared with Sally, I don't know if this is bothering anyone else, but I feel like this resuspend arrow here is kind of misaligned. So maybe what I'll do to help Sally out is kind of push this up a little bit to make sure that that's properly aligned. And let's do a little bit of more tweaking. Maybe I'll come in here and change this call out tail to a pink color to kind of match better with the overall template. And there we go. Yeah. We've collaborated and hopefully that helps Sally out with her figure.
No, yeah, definitely did. There you have it. Amazing. The final protocol figure with the collaborative effort of myself and Mina. And yeah, and that's how we recommend making these protocol figures. So here, to reiterate, are the steps that we are recommending, the 5 steps. We really recommend following these steps the next time you build a protocol figure. But yeah, with that being said, I think Mina has some more tips to share with us. So I'll hand the mic over to him.
Awesome. Thank you, Sally. Okay, so what Sally demonstrated was a really efficient process for creating protocol figures really from scratch. So now what we're going to dive into are our top 5 design principles that will really elevate the effectiveness of your protocol. So the 5 tips that we're going to be focusing on are following a simple composition. So composition would be number 1 there, ensuring appropriate text hierarchy, grouping things together where possible to create better associations, ensuring appropriate icon and text proportions, and simplifying, which includes simplifying our lines, our shapes, and our colors as well. So first, taking a look at composition, these are really the main compositions or layouts that are most effective at organizing visual information. And why that is because they really all read from left to right and or from top to bottom. And this mimics the way that we read text in general, so this should also be the way that we read visual information. So for protocol figures that have a few steps, really a simple unidirectional composition is the simplest way of organizing your figure. So you can see here, even though actually in this example there's a good number of steps, the steps, the actual visuals for them are quite narrow and they can fit very easily in just a linear fashion without needing to use extra rows to present this data. So a linear composition here works really well. For cyclical processes, what you want to make sure of is that everything is reading clockwise. And this is because we read our clocks clockwise, so it's just the natural way that we assume that cycles are going to follow. So make sure that it's clockwise, and you want to also make sure that the starting point is in the top middle or top left of the circle. So in this case, you can see that the patient is it's near the top and also towards the left side, and this is a natural starting point for your eye to kind of focus on while you go through that cycle. For protocols that may have many, many steps and you can't fit everything in just one row, we use a Z composition. Uh, and this is really most effective at organizing these figures because it retains that left to right reading order for both rows. So you can see in the top row, reading left to right, and in the bottom row, after we kind of zigzag our way back to the beginning, we're still reading from left to right, so it retains that reading order. For protocols that have converging or diverging steps, which a lot of them do, we want to use simple fork-shaped compositions that also read left to right or top to bottom. And this really helps us avoid diagonal lines. I'm sure a lot of people here have seen publications where there's a lot of diagonal lines that seem to point in many different directions, and that can cause a little bit of visual confusion. So consolidating everything into a fork like this when there's a diverging or converging step really helps organize your visual flow. So here's an example of a figure that we actually made over. So this was submitted to us, and we were tasked with creating a makeover for it. So you can see the original is on the left and the made over figure is on the right. And really what we noticed was that the original one mostly suffered from a lack of clear direction of information flow.
Oh,
So you can see in the original one on the left, my eye doesn't really know quite where to go and where to start, really. So my eye, if I'm trying to follow this composition, would zigzag all the way across the figure, kind of going up and down and tracing backwards. And then maybe I'll end up somewhere in that left bottom quadrant, but I'm not quite sure that I've read the figure correctly at all. So the arrow flow and the way that the elements are placed on the canvas really don't do a great job in guiding me as a viewer. So by comparison here, you can see that in the made over version, we've simplified the composition quite a bit so that it's linear, it's just top to bottom. And we've also employed a couple of other techniques, including groupings, which we're going to talk about later. So you can see that we've grouped things together based on association, so that left and right portion of the protocol. But the composition now is a lot easier to follow, even though all the information is retained. It's the exact same information as the original, with just a little bit of tweaking of information flow. It's a lot more effective. And there we have it. So side by side, if you can compare them, you can really clearly see how just simply restructuring your protocol using one of our recommended layouts really drastically improves both the readability and the comprehension. Okay, so let's talk about text hierarchy. So before we dive into this, actually, let's all take maybe 5 seconds to just read the information that's presented on the slide. Okay, so I love showing this because it got me the first time I saw it as well. And really what you would have likely noticed is that the text size and formatting were the main things that influenced the reading order rather than text placement per se. So you would have read the thing that's in the middle first, that's big and bold and has this black background behind it. And then you would have read the thing right underneath and the one underneath that. And then you would have went all the way back to the top with that tiny text that you may have missed originally. That would have been the last thing that you read. So Text size and formatting is often overlooked, but really this is an essential tool in conveying the intended reading order of information and the hierarchy of information that you're trying to present. So let's talk a little bit more about that. So here's an example of a figure that was submitted to us also for a makeover. And the thing that, there's a few things that we can probably change about this figure, but the thing that stood out to me at least the most was poor text hierarchy and formatting decisions. So if we go to the next slide, we actually counted a total of 4 different text sizes, but this is also in addition to different formatting choices, including bolding and italics, that didn't really seem to convey any important information. So it was a little bit all over the place. It caused a little bit of confusion in terms of what information falls under what other information. The figure also generally suffered from a little bit of over design. There was, boxes within boxes, which we tend to try to avoid. So as much as possible, we avoid creating too many boxes within boxes because it creates this kind of Russian nesting doll effect, which causes a little bit of visual confusion as well. So we'll fix that in our makeover. So here you can see this is all the same information. Everything is still retained. We've kind of taken away that Russian nesting doll situation of having too many boxes within boxes. And we've also, most importantly, simplified and really strategically use our text hierarchy in order to guide the information flow. So in total, if we can go to that next slide, we now only have 3 levels of text sizes. And this really highlights the reading order going from the title to the subtitles to the labels within the figure there. So now if we compare the before and after side by side, you can see that it's important to use font size and formatting really strategically. And as a guiding principle, the number of different sizes and formats of text should correspond exactly to the number of distinct levels of information. So think about your titles, your subtitles, your supporting text, your footnotes, and so on. That should correspond exactly to the number of different sizes and formats that you have on your figure. Okay, so now let's talk a little bit about grouping elements. So just by simply grouping things together, you can really help a figure be more easily consumed. So when I was a grad student, it always made me feel a lot better to organize my items on my bench, as you can see here, by category. And using things like trays, for example, really helped with that. And you'll notice here that, again, it's the same number of items on both benches, but just by simply grouping things together and organizing them based on use cases, everything is a lot easier to follow. I can find the tools that I need a lot more easily, and everything just makes a lot more sense. And there's less of a cognitive load for me really to be able to find what I need. So let's take a look at an example here. So again, here we have a makeover with the original being on the left and the made over figure being on the right. And in an example on the left, there's really no clear grouping of these bacterial cells that relate to each step. Yes, thank you, Sally, for zooming in. Sorry, I know it's a little bit hard to see there. Um, and there's really insufficient negative space between each of the steps. So you can see that, there's not a very clear delineation of what should be associated with what there. There's also an issue with the titles for each of the steps. So you can see, step 1, step 2, step 3. It's really hard to kind of find them because they zigzag up and down. And there's a decoupling between the actual step titles and the description for the steps, where sometimes the step title is on top and the description is on the bottom and vice versa. And it's just really difficult to kind of follow along with that. And if we scroll down to the right there, we fixed a lot of these issues with some grouping strategies here. So first we used callout circles to help visually group bacterial clusters that are associated with each other. So you can see for each step, it's much more easy to see which bacterial clusters are associated with each step. And you can see the change over time in what's happening with the bacteria there a lot more easily. We also grouped the related text with each of the descriptive steps for each one and placed them all on top. And as a best practice, it's usually good for protocols to place your step titles and descriptions at the top of each step. And then we also did something at the bottom there by adding redundant legends. And that really helps keep the steps separate. So this is not always necessary, but in this case, because there are so many different things happening with the bacteria over these multiple steps, it really helps to just highlight for each step what the legend is and what's happening inside with these bacteria. And that really helped consolidate the information as well. Awesome. Alrighty. So tip number 4, now we're going to be talking a little bit about proportions. So poor proportions can really make your protocol difficult to interpret, and this can really go in both directions. So for optimal clarity, it's important to strike that balance between emphasizing the key visual elements of your figure while also retaining sufficient padding around the elements to try to avoid crowding. So you can see in these examples, that middle example is really the one that strikes the best balance in terms of proportions, where everything is clear, but it's still spaced out enough that there's enough negative space between each of the elements. Whereas on the left, all the icons are a little bit too large. Your eye doesn't really know exactly where to focus. Everything's a little bit too close together. And on the extreme opposite side, on the right there, you can see that elements are maybe a little bit too small. It's kind of hard to make out any details. The key elements don't really stand out, so you're not really sure what to focus on. So striking that balance is really key. Now, if you're struggling with proportions in your protocol and want to include specific details without using too much space, one thing to consider is maybe using a callout. So we demoed this in a couple of figures already, but let's walk through it in a little bit more detail here. So Sally's going to help me out by actually demoing this. So here we have a dish, maybe it's an assay dish, and we want to really call out a specific area of the dish so that we can add details to it. So the first thing that we're going to do is just make a copy of this. So we're going to duplicate it, and then we're going to enlarge it to the size of the zoom that we want. That looks pretty good. Now we can go to our crop tool and just crop the area that we want to focus on for our details. Looks great. And then we're going to do something here. We're going to click on crop stroke, and we're going to add a stroke to this crop. And I'll talk about why in a second here. So let's say a stroke of 2. And if we exit out of that, you can see now that my cropped area already has a border around it. So this really saves you the step of having to create an additional box to put around the area that you're cropping out by kind of incorporating that feature into the crop tool. So that looks great. The next step here is we're just going to create that association by adding a callout tail. So we'll choose a shape, a triangle in this shape, in this case, and that triangle can act as our callout tail. We just need to make sure that it's sized appropriately and pointing to the right area that we're calling out. And then of course we can click on that shape, and in our left panel we can format it to the way that we want. So maybe we'll change the color to match something to make it match a little bit better with the template, and we'll remove that stroke on the outside of the shape because it's not necessary. And that looks pretty good there. Awesome. Now that we have this zoom out, we can add all the details that we would have wanted to add and make sure that they stand out without being too small in the original figure, but also without taking up too much space by making the original figure too large. So we're just going to focus on the zoom area, and in this case, we want to add some antibodies. So let's pull in some antibodies there and add them to the canvas. And now we can make sure that they're nice and clear. We can resize that to make it just a little bit bigger there. Perfect. And that's a really effective way of just making sure that you're saving space, but still calling out all the important detail that you want and keeping the proportions appropriate across your entire figure. Awesome. Thank you for showing us that, Sally.
No problem. I'm also still here, by the way.
Awesome. All right, so now let's talk about simplifying. So this is usually something that happens near the end of our figure making process. Maybe we've already kind of figured out our layout and our groupings look pretty good. And now we just want to kind of go back and maybe simplify some of the elements down to just make it really elegant and effective. So First, we'll talk about simplifying lines. So different styles, thicknesses, and types of lines can really negatively impact the information flow and distract from key features. So you can see in the example on the left here, a lot of different kinds of lines are used with different styles. So you have thick lines that have a fill and a border. You have really thin lines. There's a lot of different colors happening. There's just too much going on there that it becomes distracting from the actual steps that you're trying to show. The thing that we really want to keep in mind is that different types or sizes of lines should have very specific purposes and used strategically. So there are examples where we want to use different kinds of lines. For instance, we can use thicker lines to convey an increase in production, or faded arrows to convey movement, or dotted arrows to convey weaker or uncertain signals. But in general, you can see in the example on the left here, we've mostly stuck with our kind of standard blunt-ended arrows. They're thin, they're not too distracting, and we've also used that, our best practices in terms of our layouts and composition by using those forked arrows as well. But really, there's not a lot of variation in terms of the style of the arrows and the formatting of the arrows. And that really just helps keep focus on your actual steps without adding any unnecessary distraction. Now let's also talk about shapes, simplifying shapes and icons. So in general here, we want to stick to as simple of a representation as possible to avoid unnecessary complexity. So we always like to default, even as science designers, default to the simplest representation that we can get away with for any particular element. So you can see here we have representations for proteins, membranes, and nuclei, and really only step up the complexity of representation when necessary. So an example here could be maybe in your figure, the protein structure is a key element of your figure. So you need to show that protein structure in a little bit more detail, and that's perfectly fine. Or maybe your phospholipid bilayer is being disrupted in some way, so you actually want to show the phospholipid bilayer rather than just using a line, and that's perfectly appropriate as well. Or an example of the nucleus, maybe you want to show movement in or out of the nucleus, so having that representation of the perforated phospholipid bilayer there would also be useful, and you can do that as well. But in general, I would say 90% of the time, either that first level or second level of complexity works really well for representing what you need to. And we only use those more sophisticated, complex representations in very particular cases. And finally, let's talk a little bit about color. So minimizing the number of colors used to can really make a figure look a lot more harmonious. So you can see in the examples here, the one on the left uses a lot of different colors, kind of without rhyme or reason. The colors are almost chosen at random. I think we counted 15 distinct colors there, and the palette overall is not very harmonious. Whereas in the example on the right, we've stuck to a mostly harmonious color palette here of blues and purples. And what this really allows us to do is use color strategically in order to call out details that we want to call out. So here we're using that orange kind of yellowish color to just call out specific things, while everything else, we're kind of considering as similar importance. Alrighty. So here again are the 5 tips that we just covered, just as a quick reminder. Uh, and now, of course, that we're all expert level designers, what we're going to do is we're going to put our collective brains together and critique a few figures so that we can really see how to improve them and test our knowledge a little bit with what we learned. Um, so we'll do a couple where we're gonna launch into polls in just a second, and then the last example we're actually gonna break it down. Uh, we're gonna break it down into all of its components and really go step by step to figure out how we can improve it together. And of course, don't forget to stay tuned for the very end. We're gonna have a special announcement, so you definitely don't want to miss that. Alrighty,
Okay, are we ready? Should we get into it?
let's do it.
All right, let's go. Here's the first one.
Okay, so in this first one, we're going to launch a poll, and we're curious to hear what everyone thinks in terms of which of these protocols has a better layout.
We'll give it a few
Oh,
seconds for people to vote.
lots of votes coming in, and it looks so far like B is in the lead at about 78%, 22% for A, and it's kind of settling around there, about 20 to 80% split here
Mhm.
for A and B. All right. Yeah, it looks like you guys have been really paying attention. So B in this case would be the correct answer. Although A and option number A, your eye actually travels a little bit less between steps 3 and 4, as you can see there. You're just going straight down. So maybe that's why about a quarter of us or so ended up choosing option A, which totally makes sense. But what ends up happening here is once you travel from step number 3 to 4, now you're kind of forced to read backwards. So now you're having to read from right to left, and that creates a little bit of a cognitive load. And option number B, so this follows our kind of standard Z-shaped composition when we have 2 rows of information. And although your eye is traveling a little bit more between steps 3 and 4, after you get to step number 4, you're following that left to right reading order, which is a lot easier. Awesome. Really interesting results in the poll.
Mhm. Okay, next one then.
Next one. All right. So the question for this one is, which of these protocols is more clear, just in terms of providing the information that it's trying to provide? Let's see what everyone thinks here.
Oh,
Oh,
this one's a bit more neck and neck here.
a little bit more split for sure.
Yeah.
There's a clear winner so far, but it's definitely a little bit more split than the last one. I'll maybe give it a couple more seconds before revealing where we're at.
Mhm.
Okay, so it's starting to kind of settle around 65% say A, about 35% say B. So this is super interesting. And actually I think maybe the reason this one's a bit more split is there's a couple of different things going on here. So A would actually be the correct answer in this case. So a lot of people got that right. The majority got it right, which is awesome. So here in option A, it establishes a very clear reading hierarchy in terms of using text size and formatting. So we can see that the subtitles kind of stand out a lot more compared to the other text on the image. So we've established where the subtitles are and where the supporting information is. So that's really the first key difference between A and B. And then for the icon labels, um, oh, sorry. And then as an additional thing, we've also used color coding effectively to kind of differentiate the main differences between the protocol variations. So in option A, you can kind of see we've used that blue color to differentiate the main difference between the top and bottom portions of this protocol, the blue color being associated with the machine learning approach versus the more traditional approach for drug discovery there. And that really it is an effective way of using color. Although we tend to try to minimize color, this is an appropriate way of using it to really highlight the difference or the key component of your figure there.
Hmm.
Awesome. Super interesting results. I love doing these polls. So now let's dive into kind of a more step-by-step approach to critiquing one of these figures, and I'll give it away to Sally.
Yeah, nice. Yeah, so like Mina said, more of a step-by-step breakdown just to see where everyone is at in terms of understanding the tips that Mina has presented. So yeah, that'd be great to use this as a figure to sort of do that. Okay, so this figure, I will note, was submitted by a user for a figure makeover. So a few notes that I maybe want to mention before we jump into, again, polling. So I would say the user chose appropriate icons, has generally good proportions, and the symmetry in the first 2 rows um, does help guide the viewer through the figure. But of course, improvements can be made to make this an even more successful figure. So at this point, we'll throw on a poll and ask everyone which of the 5 tips, so the 5 tips are composition, text hierarchy, group elements, proportions, and simplify. Which of these can be applied here, do you think, to make this figure even better than it is now? And of course, if you have specific suggestions about how this figure can be improved, please feel free to type those into the chat as well. Would love to see them. And once we get sort of some results and some suggestions, we'll launch into step by step how we could make this even better. Yeah, so results are coming in. People are voting. Of course, they're not, the multiple tips can be applied at once. So feel free to vote for whichever you think applies here. Yeah. Let's see, give it a few minutes. People are saying alignment of elements for sure. Yeah, makes sense. Yeah, so amongst the tips here, amongst the five, it's quite evenly split, honestly. There's no one clear winner out of all of them. Someone's mentioning, yeah, Anna's mentioning the bottles are a bit confusing for sure. Text size, something could be applied there.
Yeah, some comments
Putting
about
space
spacing.
between the rows, text size and proportions. Yeah, good answers. Yeah, it makes more sense here that there's not like one tip that wins out of all of them. For sure, they can all be applied in some way or another. I think generally people are having maybe a hard time understanding or reading or finding out which way to read, like what things belong to what. I think that's been a general thing. Okay, cool. Yeah, that's some interesting results here. So I guess with that, we'll jump right into it. To be honest, yeah, I think there, like I said, even split between the five tips, and that's kind of the truth, to be honest. But for the step-by-step breakdown, let me first start off with composition. So I think a lot of people in the comments have mentioned it's kind of hard to follow the order, and some of the steps are kind of like mushing into each other. So for sure, what you can do here is simplify the layout a little bit more. So what was maybe not ideal about this figure is that there are three rows quite close in proximity to one another, which means that it's hard for you to actually read the figure. So by making this figure into more of a Z shape with two rows like this, the more the steps are linear and easier to follow. So much easier to read one and then a second row rather than three rows. So yeah, that's the first sort of improvement. Of course, that this does introduce some other issues, right? So for example, there's now quite of a imbalance at the top and the bottom. There's many more steps at the top and the bottom. So the next tip I would actually apply here is to group elements. So let me jump into how it could be grouped here. So here, what we did was actually consolidate some of the steps together. So before, the media bottles were a separate step of their own, which means they took up more space. But now we can just put the little media bottles right on top of the six well plates so that it takes up much less space. You can also group some of the information right on top of the arrow as well. So before, if I actually, let me go back to the previous one, the timings were included in this texture, and so it kind of was a bit harder to understand. If you group them on top of the arrows, much more clean, and it's also easier to understand as well. So some strategic grouping to give some more space, more balance here. Another thing you could do here is to simplify. So for example, there are a lot of colors going on here. So you can make this color much more closer to this media bottle and vice versa. Because right now it's sort of introducing inconsistencies and therefore a little bit of confusion as well and a lot of visual clutter. So for example, I can make this a darker red like this, for example, and I can make this more pink, and I could do that change here as well. I also do notice that the arrows are different in terms of styling as well. So this is a blunt-ended arrow. As you could see, if I go to the left here, there's no effect. But this one, if I click, there is a line fade. So can make everything more simplified and remove the fade in there to make it a little bit more streamlined, a little bit more consistent amongst the arrows. And so once you apply some of the simplification steps, you can land on something that looks like this. There you go. And with that, you should always do it before and after, right? To compare where we were at in the beginning with where we ended up at the end.
Awesome. Thank you, Sally. And we had a couple of comments actually that were really interesting in the chat about kind of the order of the steps.
Hmm.
It seemed like we changed it from the media pointing, or sorry, the plates pointing to the media to the media
Hmm.
pointing to the plates, and there's a little bit of confusion there. So maybe one thing that we didn't mention in this particular example is that there were also some visual inaccuracies with the original that we ended up confirming with the creator. Indeed, the media is supposed to be being added to the plates, and the timing steps is are in between each of those additions. So just wanted to clarify that. Of course, we also always want to make sure that scientific accuracy is paramount, so that takes precedent over kind of any artistic direction. So we just want to make sure that
Mhm.
that we take that into consideration as well,
Yeah, 100%. Would rather go choose scientific accuracy over design, if you will.
for sure.
Yeah.
Okay, awesome. Thank you so much for showing us that, Sally. And now that Sally has her polished protocol ready, maybe she wants to present this figure to her teammates in a PowerPoint presentation. So what this typically would mean is that she would have to switch multiple times between platforms. She would have to maybe copy and paste or import static versions of her figure from BioRender into PowerPoint. And if she wants to make any changes to her figure later on, she would have to kind of break that workflow in order to do that, and then re-import or re-copy and paste her updated figure into PowerPoint. So we are super excited to announce that we have just eliminated
Woohoo.
all of these pain points and all these time-consuming steps, and ByRender now has seamless PowerPoint integration. So we think this is going to be a huge game changer for all of the scientists on this platform. It's something that's been requested for a very long time. It obviously came with a lot of technical challenges, but we're really happy to present it here. And Sally will take us through her workflow now that she's created this figure and she wants to kind of put it in PowerPoint. We'll see a little bit of a demo of how that works.
Let me just share my new screen so people can see. Oh,
Awesome.
people can see my screen now?
Yep, looks great.
All right. Awesome. Great. Um, like Mina said, we now have a nice BioRender add-on to the PowerPoint. So for the purpose of this demo, let's say this is a PowerPoint that I'm working on to update everyone on the status of my Scratch essay, the figure that I was showing before. So I have the slides now formatted in a way that my institution wants to, with a nice green background. I have all these layouts prepared. I just need to add my BioRender figures to it. So to do that, once I have the add-on installed, I already have it installed, I will have this handy BioRender button at the top right-hand corner. I can then click on it and click on Add File to access all my recently opened BioRender files. So here they are, and click on the one that I actually like to add, which is this one in this case. So after a second, it should add on my figure. So you can see that once it's been added in, I can resize as much as I need to, and I want to, without sacrificing quality or pixelating the image, since I have used the add-on. But now you'll notice that since I have this little white background on, it does obscure the nice green background that my institution wants me to use. So what I can do at this point is toggle on the transparent background, like so. I can also toggle off this collaboration link. Although it might be useful, it might be distracting during a presentation, so I can turn that off. I also noticed that maybe I want to change some things around. Maybe for some reason I want to make this arrow bold. What I can do in that case is I can select on the figure and then click Edit in BioRender. What that will do is open the figure in BioRender directly. And then I can make the change as I need to. So I will make the change that I talked about, make this a bit bolder, like that. Once that change has been made, if I go back to PowerPoint, you'll notice that that has been reflected here and it has been noticed basically. So I can go ahead and update image like that to make the arrow bolder, like so. So yeah, seamless integration, translation between the BioRender file and the PowerPoint slide. There you go. I will say
Amazing.
that, yeah,
We're
thanks.
getting a lot of great reactions in the chat. Everyone's super excited about this.
Nice. Awesome. Awesome to hear. Yeah, I will say that making sure that this integration between BioRender PowerPoint as seamless and easy to use as much as possible was definitely not definitely technically challenging, but we understood that PowerPoint obviously is a big part of a lot of our user science communication workflows. So yeah, we made sure to develop this and add it into to BioRender to streamline everyone's workflow.
Awesome. Thank you so much for doing that for us, Sally. And I've dropped the link to the help article on how to get PowerPoint integration in the chat there. So hopefully a lot of folks here can make good use of it. And with that, we're coming up to the end of our presentation here. So if you have any questions about how to use a feature or different licensing types, you can always visit us at help.biorender.com with lots of great step-by-steps and how-tos that are covered in our help docs. You can also contact us at support@biorender.com. We will be sending a link of this recording as well to you so that you're able to watch it at any time in case you wanted to review any of this material. And we're also launching just a quick feedback poll here. If you can take a quick second before exiting and fill that in for us, that'd be really, really appreciated. And as everyone is doing that, I'll just mention that for any information on institutional pricing, you can visit byrender.com/contact and complete the form there. If you indicated that you'd like to speak with our sales team when you register for this webinar, someone on the Byrender team will already be reaching out to you after this webinar. So if you'd like to request a demo for your team or learn more about your our institutional licensing options, you can let them know directly. Yeah, and thank you so much for everyone who's participating in the poll right now and for our feedback. And really appreciated everyone making the time to join us today. We had a great time. Really happy to see the great engagement in the chat and everyone being super excited about PowerPoint integration. We
Yeah.
hope that everyone here learned a lot about how to create really powerful, impactful, and effective protocol figures. Hopefully, you can take that learning into our publications and presentations. And yeah, thank you so much. We'll just hang out maybe for a few more seconds while people finish filling out those that feedback pool. Thank you.